If individual cells of a multicelled organism are not genetically identical, then the reproductive success of one cell does not automatically imply the reproductive success of other cells. So if one genotype is successful, a different genotype is not therefore also successful as a consequence of inclusive fitness. What you have instead is the potential for competition between different cells. If the cells are genetically identical, at least from an evolutionary sense, there cannot be competition between the cells because there's nothing for selection to differentiate. But once cells are different from one another, or once organisms are different from one another, once there are different alleles found at a given locus, then you have the potential for natural selection to act. Such situations destabilize cooperative behavior between cells. So ideally, if you're going to have a multicellular organism, that multicellular organism is not going to have genetic variation within it. An extreme example is a lack of cooperation displayed by pathogens or parasites towards an unrelated host. So what is a parasite? What is a pathogen? It is a different genotype that has found itself intentionally in the midst of a genotype that is not identical to it. And so as a consequence, there's genetic variation. Now, technically, this is not natural selection because we're talking about two different species at this point, but nevertheless, there is a conflict that exists because you've got two different genotypes occupying the same space, and those genotypes are at least potentially going to be in competition with one another or in conflict with one another. And in the case of parasites or pathogens, it's in conflict. And the same is to a degree true even if the individuals are the same species but differ in their genotype. If they are occupying the same location, they are going to be in competition with one another and potentially in conflict with one another. Natural selection is going to be able to distinguish between the two different genotypes. So once you have different genotypes, suddenly you have the potential for natural selection creating a destabilization of, for example, the potential for cooperative behavior between individuals. Multicelled organisms have evolved a variety of mechanisms that cause genetically identical cells to act cooperatively. So it's best that we start with genetically identical cells, but there are different layers that are built upon that that assure that cooperation continues into the future. They also have evolved mechanisms that serve to prevent non-genetically identical cells from acting not cooperatively. Finally, we have evolved mechanisms that serve to prevent non-genetically identical cells from arising or invading at all. The key is to keep the cells as genetically identical as we can, and to also assure that otherwise, regardless, the cells have incentive to act cooperatively. So these various counter-defection mechanisms include individual cell replicant restraint or replicative restraint. So what this means is that the cells impose upon themselves a restraint on their own replication. They are attempting to be economical rather than expedient. They try to be cooperators rather than defectors. And this comes about as a consequence of their own genes acting upon their own phenotype, causing those cells to display restraint, for example, in their population growth. We also have mechanisms that combat mutation. We talked about this way back when at the beginning of the term. Proofreading enzymes, for example, are a means by which cells reduce their own mutation rate. We start off babies with one or at best a few cells. Typically it's one cell. We have two gametes that fuse and we form a single cell, a zygote. And then that zygote via asexual replication, that is mitosis, in eukaryotes and binary fission in bacteria, then give rise to a multi-celled individual that, except for mutation or invasion by parasites, is genetically homogeneous. We also have immune systems, and these control our own body cells. So if our own body cells go rogue, start down the path towards forming tumors or cancer cells, we have means of interfering with that progress. And of course, we also use our immune system to minimize the potential for cheaters from without, for parasites to be able to take advantage of our otherwise cooperating cells, to take advantage of the functionality of our bodies towards their own end rather than our own reproductive potential. And then finally, we have a requirement for functionality in baby-generating tissue. And this is a way of effectively testing the waters, the genetic waters, prior to gamete production, or even following gamete production. We have basically set things up so that they're not as cushy as they possibly could be for our cells and our gametes and our zygotes and our offspring. So that these offspring have to struggle at least a little bit and thereby prove their worth before they can continue on in their development, and as a consequence use up more of our resources. A great number of the embryos that form within women's bodies, I can't say my own body, do not make it through to the point of their full gestation and then birth. And this is because they are not viable for various reasons. And one way of interpreting that is that the lack of viability represents some sort of defect on their part that interferes with their ability to continue onward to the point of exiting the mother's body. And as a consequence of that, we are able to cut our losses early on. And in fact, the number of fertilized eggs that don't survive presumably is much larger than the number of embryos that don't survive. And certainly the number of gametes that don't survive is vastly greater than the number of fertilized eggs that don't survive. And in fact, starting babies from only a single cell actually serves as a cancer-fighting strategy because we're eliminating our defectors from the start. And in fact, helps explain to some degree why older people are typically more prone to cancer than are younger people. We don't make our babies starting with somatic cells that have become cancerous. Instead, we do everything we can to make sure that our offspring are as genetically intact, as genetically effective as we can succeed in doing. To initiate the next generation, we bottleneck it so that the individuals that result are the product of just a single cell. And if that cell has a genotype that can give rise to a high level of functionality and future reproductive success, then they will be more likely to be evolutionarily successful. And if the genotype of those cells do not give rise to a high level of functionality or potential for reproductive success, then you will see a loss of those genotypes from the population.